Well, good afternoon, everyone. I'm Doug Petke, Department Head of Physics, and I'd like to welcome you to the Arts and Sciences Week. This is going to be the best part of the week, but there's going to be plenty of other great activities during the rest of the week. And so it's my great pleasure to introduce the Arts and Sciences Student Lightning Talks. From the time the students cross the Earl Bridge that you see in the background, before the first night of classes, to the time they cross it again at graduation, WPI's programs allow students to imagine, innovate, integrate to make an impact in the world. Each student's path is unique and made possible by a dynamic and creative environment that we have at WPI, and that's made possible through many generous sponsors, as well as that fund a wide range of programs, from the early research experience in e-term, from summer training in the arts and sciences research, the STAR program, IQPs, MQPs, as well as a lot of students that work on externally sponsored projects from the NSF, the NIH, NASA, and quite a few other ones. Working in these programs prepares students for the next stage in their career, gets a significant head start, whether it be employment, starting a company, or going to graduate school. These 11 students represent 9 projects and are excited to tell us about the significant advances they made over the past year in a diverse set of fields that's represented by arts and sciences. And so without further ado, I would like to get the students started. And what we're going to do is the students, we're going to do this sequentially. I'll start off by introducing Katie Donovan, and then as we progress through, each student will introduce the next student in the talks. Great. So, Katie?
All right, thanks, Doug. Hi, my name is Katie Donovan. I'm a senior studying biochemistry, and I've been involved in research at WPI since I was a sophomore, when I began volunteering in Dr. Carissa Olson's lab over at Gateway. And for my MQP, I've actually continued in the Olson lab, and in my project, I've been investigating the link between Parkinson's disease and lipids. Next slide. So most of you have probably already heard of Parkinson's. It's the second most common neurodegenerative disease, and it's a really important area of research because there's currently no cure for Parkinson's and no great way to diagnose Parkinson's early on. And this is simply because we don't know what causes Parkinson's. There are a handful of genetic mutations that are linked to familial Parkinson's disease, but most cases of Parkinson's are sporadic and they occur later in life, and we don't know why. So relevant to my project is this fact that Parkinson's has been linked to dysregulated lipid metabolism. So what are lipids, you may be wondering? You're probably more familiar with lipids than you think because they're what we refer to in our diet as fat. So the most common lipid in your diet is this triacyl glycerol shown on the left, and it's found in butter and oil. But the type of lipid that I have been studying in my MQP is this class of lipid called phospholipids on the right, and these are abundant in our cellular membranes. So our cells need a barrier to distinguish between their external environment and their internal environment. So it's really important to have a healthy phospholipid composition to have a healthy cell. Next slide. So in the Olson lab, we're able to study lipids using the model organism C. elegans. So as you can see, C. elegans are a tiny worm. They're about 1 millimeter long, and they're found in soil, but they're found very commonly in laboratories because you may be surprised to find out that the genome of C. elegans overlaps pretty highly with the human genome. So they're a great research tool. They only live about 2 weeks and they're easy to maintain. So in my MQP, I have been looking at this one C. elegans worm that has a mutation in the gene UNC26. And this gene is the same gene that in humans we refer to as PARK20. So a mutation in this gene causes early onset Parkinson's. And you can see that the worm, the UNC26 worm, looks a little bit different than the worm on the left, which is the normal or wild type worm. So the UNC26 worm actually tends to move in circles, and it has delayed development and is a bit smaller. So in studying the lipid composition of the worm, I found that there's an increase in this one lipid species called PEs in the UNC26 worm. Why do we care about this? Well, changes in lipid composition are a potential way to diagnose Parkinson's, so they could be a biomarker. And also, you may have heard of lipid dietary interventions, such as when people tell you to eat more omega-3s or eat less saturated fat. So that's a type of lipid dietary intervention. So maybe there is a lipid dietary intervention that could be useful in treating Parkinson's. So my research is a really early step, but I hope that it ultimately contributes to the body of knowledge that will help us diagnose and treat Parkinson's better. And so thank you. With that, I'm going to turn it over to Sarah Tarantino to talk next.
Thanks, Katie. So hello, everyone. My name is Sarah Tarantino, and I'm a biology and biotechnology major here at WPI. The project I'm talking about today was actually inspired by a project I completed this summer through the Arts and Sciences STAR program. My experience in STARS led me to the development of my MQP topic and the work I'm doing in the Srinivasan lab. So today I'm going to talk about some of the research I've been doing, looking at the interactions between the gut microbiome and the progression of Alzheimer's disease. So these may sound like two completely unrelated things, but I'm hoping that by the end of my talk, you'll see how our gut health can directly influence our brain. Next slide, please. So our gut microbiome is composed of over 1,000 different microbial species living in our gastrointestinal tract. These microbial cells actually outnumber our own cells in a ratio of 10 to 1. The microbes in our gut communicate with our brain through a signaling pathway known as the gut-brain axis. This is a bidirectional communication pathway between our gut and the central nervous system. It utilizes neural, hormonal, and immunological signaling. As seen in this diagram here, the brain communicates with the microbes in our gut to help regulate GI function. And the microbes in turn communicate with our brain, providing different metabolites and neurotransmitters that can be used elsewhere in the body, as well as sharing sensory information so we can keep track of the conditions of the GI tract. However, the success of this pathway is dependent on the different types of microbiomes that are living in our tract and in what amounts they are living there. So disruption in this healthy gut microbiome has been linked to several neurological disorders, such as anxiety and depression, and also neurodegenerative disorders like Parkinson's disease, multiple sclerosis, and what I'm looking at, Alzheimer's disease. You go to the next slide. Alzheimer's disease is the most common form of dementia, and it's the 6th leading cause of death in the United States, and currently there's no cure. The understanding of Alzheimer's disease is that it's caused by a buildup of a protein called amyloid beta in the brain. These amyloid beta proteins will aggregate together and form these masses of protein known as plaques. The plaques are harmful to the brain, and they cause neuro impairment and cognitive impairment by blocking nerve signaling. So as seen here in the picture to the right, there's a greater amount of amyloid beta buildup in an individual with Alzheimer's compared to a healthy model. Through recent research, there's been found that Alzheimer's disease patients actually have a disrupted gut microbiome, and this is seen in the diagram to the left. It is believed that the disrupted gut microbiome may contribute to the formation of amyloid plaques in the brain. So it's possible that the amyloid protein these bacteria use to communicate day to day may accidentally cross the blood-brain barrier and start the formation of the plaques in our brain. You can click once more. So in my research on this topic, I've been using the model organism C. elegans, and as Katie already introduced, C. elegans are a great model organism. They have many similarities to humans on a genetic and neural level. Specifically, I've been using a strain that expresses human amyloid beta protein, so it acts as an Alzheimer's disease mutant model. In my research, I'm looking at the differences in the gut microbiome composition between the healthy controls and the Alzheimer's disease model. I use behavioral assays to track disease progression and DNA sequencing of the microbiome to characterize the different microbes present. Overall, I'm looking to see if we can use C. elegans as a model organism in this context. Having a simple model organism such as C. elegans for this context would be really useful in future research, such as investigating if there are specific microbes or community of microbes that are involved in the amyloid deposition, or finding ways to reconstitute the gut microbiome that is disrupted in diseases such as Alzheimer's. It is much easier to modify the gut microbiome than develop drugs that can target the brain. And modification of the gut microbiome through simple interventions, such as food-based therapy or probiotic supplementation, may create new preventative and therapeutic options for Alzheimer's disease. So thank you so much for listening to my talk today. I would now like to turn it over to Justin.
Hello, everybody. So my name is Justin Pulcari, and I'm a senior undergraduate student working in the King Lab here at WPI. And today I'll be presenting results from our mindfulness-based blood pressure intervention for hypertension, more commonly called high blood pressure. So mindfulness-based blood pressure reduction programs have been used to evaluate the effects of self-regulation mechanisms in blood pressure determinants. And MBBP reduction coincides directly with improving cardiovascular health and shows promising results in bettering the lives of patients with hypertension. So in the general population, mindfulness promotes the practice of good physical and mental health and is designed to deliberately focus a person's attention on the present experience. With regards to MBBP intervention, emphasis is put on reducing blood pressure in participants. So a quick background on hypertension. Hypertension is considered systolic blood pressure greater than 130 and diastolic blood pressure greater than 80, and has a prevalence among adults of 31.1%. Hypertension is often comorbid with anxiety and depression, and MBBP has previously been shown to be effective at decreasing blood pressure compared to controls. So for our study, about 150 subjects were involved initially. We ended up having 12 subjects in the MBBP intervention group that participated in the 8-week intervention program and 24 subjects in the control group. The difference in group sizes was due to COVID-19, where it lost an imaging cohort that was mostly MBBP subjects. Using diffuser tensor imaging MRI, we were able to investigate the neural substrates of MBBP and their relationships to clinical metrics. We found that MBBP increased mindfulness and decreased depression scores with a trend for decreased anxiety. It's worth noting this trend due to the comorbidity of anxiety with depression. MBBP altered DTI tracts in several limbic and hypertension-related neural tracts, including the right cortical spinal tract, left superior corona radiata, right and left cingulum, the right fornix and stria terminalis, and the right and left superior cerebellar peduncle. Changes in these DTI tracts were associated with mindfulness and depression, as well as blood pressure and emotional regulation, as seen in the clinical and DTI correlation table in the top right. MBBP induced changes in structural brain connectivity measures, which may mediate improvements in mindfulness, depression, emotional regulation, and blood pressure. Thank you. Up next is Megan Vardy from the physics department.
Thank you, Justin. I'm Megan. I'm a senior BSMS student in the physics department. I've been working with Professor Kunta Wu for about 3 years, and I've done projects funded by STAR and the Claire Booth Luce Research Fellowship. And today I'm going to be talking all about my current project, which is on mixing. So mixing is a very fundamental process to our everyday lives. In fact, it's probably one of the first things you do in the morning when you wake up and make yourself a cup of coffee. Mixing is the combination of 2 or more materials into one. So if you take your coffee and you add some sugar and milk, you can mix these 3 ingredients into one latte. Mixing is dependent on a property called the Reynolds number. High Reynolds numbers are characterized by turbulent fluid flow, which looks chaotic, like the river flowing on the left. Lower Reynolds numbers are characterized by laminar flow, which is slower and more ordered, such as honey falling from a bottle. Turbulent fluid flow can be harnessed for efficient mixing. If you visualize dumping some glitter into this river, you can imagine that the glitter particles are very quickly going to spread out and mix within this water. For a given fluid, the Reynolds number is proportional to the speed of the fluid flow and the size of the sample. So it seems trivial that if we want to increase the mixing rate, We can take a really large volume of a fluid, add it to an industrial mixing tank, and then just stir it really fast. This works for some applications, but doesn't work for other applications that are tied to a much smaller scale. Think about the tip of a 3D printer head or a blood sample in a lab where you have a very limited quantity. How then can we increase the mixing rate when we are tied to the low Reynolds number regime? Next slide, please. I propose using an in vitro active matter system. This active matter system is composed of short, rigid rods called microtubules. I can form bundles out of these individual microtubules by adding a polymer. Then kinesin motors will bridge these microtubules and walk along them, sliding them apart. If a lot of the microtubules in the bundle slide apart, the entire bundle is going to begin to extend. The bundle will extend until eventually it will begin to bend, buckle, and eventually break apart due to the stress of the fluid. The depletion forces will then cause new bundles to form, and this extension and buckling will repeat. So how does this help with mixing? Well, imagine that you're making a bread dough. You're going to knead the dough by stretching it out and then folding it back together, stretching it out and then folding it back together, and repeating this until your dough is smooth and homogeneous. This extension and buckling that we see in the microtubule active fluids is very similar to the stretching and folding that we need in order for mixing. We often refer to these active fluids as self-mixing fluids. Next slide, please. To study how these active fluids can mix, I designed an experiment with an active fluid pipetted into a flow channel right next to a passive fluid. Over time, we can see this active fluid expanding into the passive fluid as it mixes. We can add tracer particles to this active fluid and see the same phenomenon happening, where the tracer particles are moved faster by the active fluid than they are moved by diffusion in the passive fluid. Using these tracer particles, we can measure the location of the interface and how quickly it migrates over time. This correlates to how quickly the active fluid can mix with the passive fluid. We can then vary different parameters to see how this activity of the active fluid varies the mixing rate of the fluid. And this will give insights into how quickly the microtubule-based active fluids can mix with a passive fluid and if it can increase the mixing rate beyond that of diffusion, which is often seen on the micro scale. This is crucial for applications in microfluidics, such as 3D printers or biomedical assays, which need quick mixing that is currently not possible. Thank you. And next, I'm going to introduce Seneca. Thank you.
Hi, my name is Seneca Warren. I spent my first 3 years at WPI as a civil engineer and then decided to tailor my degree to focus on the intersection between design and social and environmental justice. To support this theme, I've been working with Professor Kruger on research regarding the challenges in performing sustainable development in sub-Saharan Africa. Here's what I've learned. Let's start with a question for everyone watching. Which of these buildings do you think is more sustainable? I'm sure you know why I'm asking. It's easy to imagine the one on the left here, which is the modern one with solar panels, living walls, and a glass facade as being green. But what if we imagined that this straw dome is green? Because it is. In fact, it might be greener. When we look at the high-tech, innovative building, we think about eco-friendly materials, energy efficiency, and a low carbon footprint. But contrasted with this second structure, we can start to appreciate some other factors, like manufacturing of the materials, construction of the building, impact on the surrounding area, and adaptability of the design to climate change. In fact, many of these considerations are intrinsic to Indigenous thinking and design. This case study of green buildings illustrates how Western thought has dominated the thinking around sustainability in sub-Saharan Africa. This is because we have traditionally not looked at African ways of living as sustainable or green, because we have been conditioned to think of them as primitive or low tech. I'm not here to put down scientific thinking. Science is an incredible tool that helps fuel innovation. However, we, the global North, have an unfortunate tendency to approach places like Africa with the mindset that we need to bring our own innovations to solve their problems, as though they lack ideas or intelligence. We've neglected to acknowledge and tap into the existing capacity for innovation. Next slide. The obstacles created by scientific and Western knowledge systems in an African context are mainly due to the conventional design approach. Can you believe that only half of the World Bank's privately funded projects in Africa are actually successful? There are two main problems here. The first is a lack of outreach and therefore participation and buy-in from the local people. And the second is that the project developers have missed an opportunity to tap into a powerful source of creativity. Innovation can only get us so far, especially when conceived by those from vastly different backgrounds and cultures than the people that will end up living with these solutions. Knowledge held by those living closest to the problem need to be a part of designing solutions. Incorporating Indigenous knowledge into problem solving can help inform unique and sustainable solutions tailored to the location in which they are needed. The San people of the Kalahari Desert, for example, have been adapting to constant and drastic changes in desert climate for centuries. Their consumption patterns are refined in order to minimize impact on their surroundings, and they construct temporary dwellings that are resilient and climate adaptable. Or look to Ghana, where the people living around an e-waste dump called Agbagbloshi have created an entire economy mining this e-waste to sell valuable materials. See, there are no shortages of creative, adaptable, and sustainable ideas. We only need to access what already exists. This leaves the question, how do we perform sustainable development in sub-Saharan Africa? Next slide. For sub-Saharan Africa to meet the demands of sustainable cities, social and cultural challenges can and must be overcome. So with this new perspective, how do we weigh Indigenous ideas as equal to the cutting-edge science we can bring to design and development? African Indigenous knowledge embodies sustainability, and in order to move forward, local perspectives not only need a seat at the table, but one of equal weight to all stakeholders. There are three main models to execute participatory design that best apply to international solutions. On one side of the scale, there is end user design. On the opposite side is user-centered design. And the sweet spot is called peer-to-peer design, where everyone is equal and collaborating together. This approach eliminates many of the challenges to integration of Indigenous knowledge in modern solutions, because all stakeholders are considered peers on a just and equal playing field. Next slide. So yes, if you're picking up what I'm putting down, the real problem is us. All of us students soon to be practitioners, or at least the approach we're taking. Ultimately, while my own research focuses on the situation in sub-Saharan Africa, this is not just important for those who want to study development in Africa. This is a lesson for anybody who is going to be a professional in an applied STEM-related field. If you take one thing away from my talk today, remember that no matter where our professional careers take us, whether somewhere in the US or far across the globe, we need to look for solutions in places where we weren't trained to find them, and we must approach our work with openness and humility. Thank you. I'd like to introduce Brady from physics.
Thank you, Seneca. My name is Brady Zhang. I'm a junior physics major, minor in computer science and ECE. Before I start my talk, I would like to thank WPI's Arts and Sciences Advisory Board for their generous support of this research under the STAR program. I would also like to thank my advisor, Professor Doug Petke, for his thoughtful guidance throughout the project. This research would not have been possible without them. So on the left of your screen is a moth eye. The picture on the right represents a smart world of the future where everything is connected to each other. So what do these two images have in common? Well, the answer is anti-reflection coding of electromagnetic waves. We experience light reflections every single day, whether it be reflections of the sun on the water or the glare on your glasses. They create distractions to our everyday lives and impede our ability to clearly observe the world around us. A moth's eye structure is an array of protuberances of specific dimensions that reduce the amount of light reflected by the surface. As we develop next generation of 5G and 6G wireless infrastructure, we will move into higher frequency terahertz spectrum between the infrared and microwave bands. Therefore, the development of an anti-reflection coating in the terahertz band to maximize power and efficiency can make an impact on the development of these technologies. Next slide, please. For our project, we conducted 20 COMSOL simulations of different anti-reflection structures. These are physics simulations that help us understand the physics of different structures before we test them in the lab. Because we were interested in anti-reflection performance across the terahertz spectrum, we designed something called a sub-wavelength grating. The refractive index of a material measures how much the light slows down as it goes through the material. The closer the refractive index between the materials, the lower the amount of reflection. By creating this grating structure, we were able to gradually match the refractive indices between the different mediums and reduce the amount of light reflected by the surface. We performed these simulations for varying depth, width, and number of grooves, and picked out our best models and prepared these models for 3D nanoprinting in the lab. Due to COVID-19 restrictions, we weren't able to print these structures. However, I'm planning on continuing this research as part of my NQP project as we start using the newly opened photonics laboratory. We plan on using the 3D printed structures and testing them on our frequency domain terahertz platform. Next slide, please. So what is the impact of this research? Let's go back to what we discussed on our first slide. The future of communication of higher frequency 5G and 6G in a more congested radio space is coming. To make these technologies as efficient as possible, a quality anti-reflection coating for this spectrum becomes very important. The applications of this high bandwidth are endless. We could see a network of self-driving cars communicating with each other, making traffic of cars much more efficient, so we no longer have to sit in traffic during our commute. Or we could see a holographic communication platform that allows us to conduct meetings virtually without the impacts of Zoom fatigue that we're all now familiar with. My hope is that with continued research, we can build an exciting future of a smart world. Thank you. I would like to now introduce your next speaker, Ma.
Thank you, Brady. Hello, everyone. My name is Zhifei Ma, preferred name Mona, a junior major in mathematical science and computer science. First, I want to thank the School of Arts and Sciences for funding the 2020 Summer Training in Arts and Sciences Research Start Program and approving my individual research project. Since COVID-19 started, I have imagined the day that we don't need to wear masks anymore. We don't need to keep social distance anymore. We don't need to line up at Harrington Auditorium and blow our noses so hard, but still can't feel any nasal mucus when rotating the swab. That was one of the most awkward things happened to me last year. Maybe it also happened to you. So I was thinking, as undergraduate students, what else we can do to help protecting each other during this global pandemic? I decided to make a prediction on infected population using what I learned in math and CS courses. Here comes the SR model, a set of differential equations representing change within populations between three categories. S represents susceptible population, I means infected population, and R illustrates people who recover from and immune to the virus. I started with theoretical analysis, playing with equations and reading newly published research papers every day to keep up with all kinds of predictions made by scientists. Integrating other suggestions was important to me. I encountered a great question when presenting in the summer research workshop. Is data published by CDC reliable? The officially published data can be regarded as a kind of best case scenario, while some research suggested the actual number may be 10.5 times higher, which could be considered as worst case scenario in real life. Therefore, I ran experiments based on both data sets. After that, I did parameter study through numerical approach by using public health data of Worcester County. The most important input was the basic reproduction number, which greatly influenced the maximum number of infected populations at a time, a key to flatten the curve. Besides testing a constant basic reproduction number, I also tried on varying values over time. An SEIR model is SR model with an exposed population added, representing the incubation period. And we can further develop it into SEIRS model that takes loss of immunity into consideration. One of the major conclusions of my research explains why we need vaccinations. A basic SR model assumes recovered population will never be infected again. But in reality, a research published on February this year found that natural immunity for COVID-19 can last as long as 8 months. So the model at the right-hand side shows more accurate pattern because there's a chance that recovered people lose their natural immunity to become susceptible, and it's possible that they may get infected the second time. Therefore, the only way to completely end this pandemic is that the majority gets vaccines, excluding those who have severe allergic reactions. So if you are healthy and eligible to pre-register an appointment, please do consider getting vaccinated, not only for yourself, but also for your community. The more people are vaccinated, the faster we will return to our normal routine. I'm looking forward to the day that we will return Harrington Auditorium to sports clubs and robotics competition. Thank you for listening. And I would like to introduce the next speaker, Derek.
Thank you very much. Hello, everyone. My name is Derek Byrne. I'm an electrical computer engineering major. And I, along with my partners, Ryan Lamarche and Michael Osei, both computer science majors, are excited to bring you SentryEye, a smart and versatile vehicle security device designed to protect you and your car from anywhere. SentryEye is an MQP that was born out of last year's ECE 2799 electrical and computer engineering design class, where the task for students that year was to create a device that promoted on-campus safety and security. In conducting our research, we found that theft involving vehicles comprised much of the crime surrounding WPI's campus, which acted as a threat to student safety and security. Next slide, please. This map alone displays the thefts of or from vehicles in the area surrounding WPI's campus in the past month alone. Since then, the pandemic has only made things worse by now causing driving in the United States to go down 41% from pre-pandemic levels. So students on or even those working from home now go longer periods of time without checking on their car, parked on side streets or in parking garages, leaving them prone to theft without the vehicle owner knowing for quite some time. So to address this prevalent issue, SentryEye acts as a small modular device with a motion sensing camera that can be mounted anywhere within the vehicle using adhesive strips. Via the Cloud Companion app that both Ryan and Michael have developed, the SentryEye dashboard allows the user to control the photo and video recording, auditory alarms, satellite GPS tracking, and motion detection settings all built into the device to alert you of a break-in from anywhere. Next slide, please. Pictures of the perpetrator's face are then sent to your dashboard along with a map of where the incident occurred, which provides valuable evidence in case of a break-in. This dashboard, as seen in the picture below, can be accessed from any device with an internet connection, no matter where you are. As SentryEye is a modular approach to as a service, this means that you can add more than one device whenever you'd like. So registering a new SentryEye device is easy with its user-friendly onboarding experience. It simply uses a unique registration code used to add a new device to your dashboard without any complicated setup process. This device is designed with both affordability and flexibility in mind. We wanted it to be affordable for students, especially. So we made the device within a price point of around $50, which provides a reasonable alternative to any security competitor. As well, for $5 a month, the SentryEye service allows 4G cellular connectivity. So you're always connected wherever you can make a call in the case that your car is parked on a place without Wi-Fi or other connected services. Our market research shows that dash cams and other alternative security devices are far too expensive and lack internet connectivity as well as tamper-proof features. These competitors either lack the fail-safes needed, like sending the photos remotely in the case that the device is either moved or stolen from the vehicle, or lack basic security features like the motion detection that SentryEye offers. SentryEye will continue to perform even when unplugged with its long-lasting backup battery and will ensure that the photos are sent immediately despite any interruption in which someone pulled the cables out or anything else. If the car is even parked near a Wi-Fi hotspot, like outside of your garage, it will prioritize the Wi-Fi so that it's not using any of your cellular data as well. So these type of essential features provide the advantages needed to maintain a strong foothold in the security device market. Research into prevalent issues like this often leads to the creation of new products and services that promote innovation when done in a responsible manner. We hope that SentryEye acts as a testament to the value research can lend to new products and services, as well as ideas that will change our society for the better. Thank you all very much for your time, and I'd like to thank Professor Stephen Batar and Professor Joshua Cuneo for all their help in advising our project. And please welcome our next guest speaker, Isabel Chen.
Hi there, I'm Isabel Chan. I'm going to be talking about some work that I did this past summer with the Comet Lab here at WPI. This research was funded by the NIH, and some of this work is currently being used for MQP and graduate student research. So currently there are two main ways of treating benign and pre-malignant tumors and lesions on the vocal folds. First is going into the operating room, going under general anesthesia and receiving a general resection. And the other way is going into the office. And the way that they do this is that an endoscope is passed through the nostril of the patient, and an optical fiber is passed through that endoscope. And the fiber will fire a laser upon the tumor, cauterizing it. And this is great because in-office procedures are generally cheaper, they're faster, and it only requires a mild sedative compared to general anesthesia, making it more accessible to people who might not be able to receive that general anesthesia. However, it is quite limited still, the pool of people who can receive this treatment. One of the reasons being that it is very limited in its reach. So the laser fiber isn't always able to affect the areas that we want it to. Like the lesion might be located on below the free edge of the vocal fold, which is pretty difficult to reach, especially considering that the laser fiber can only fire along its line of sight or along its longitudinal axis. So we decided to take a look at other biomedical applications that use custom fiber tips that were angled and try to see if we could use side-looking fibers in our application. So on the next slide, what we can see is how we set up our simulation study. What we did was we created a 3D model of the endoscope, the laser fiber, and then deployed it into a model of a larynx. And then what we did was we sent the laser fiber to 10,000 different reachable points within the larynx using a motion planning algorithm called rapidly exploring random trees. From each of those points, we simulated the laser firing or the laser emission from each of those points. And then from there, we were able to see which parts of the larynx we could affect. And then on the next slide, we can see some of the results that we found. And we were able to see that side firing tips can treat more tissue area than forward-looking fibers. This is especially true for the 90-degree fibers, or the ones that fired perpendicularly. So this is really great to see. And then another thing that we found in the study was that there's a gap in which the optical fiber is unable to physically reach. And this is especially important when looking at forward-looking fibers, because this creates a gap in which the laser can't treat. And this is pretty consistent with existing medical literature and reports where surgeons have a difficult time trying to treat or access that right side of the vocal fold. But when using side-looking fibers, even for the 45-degree one, this gap that was overcome, and we were able to reach the areas that we needed to. And so we hope that by introducing a side firing tip into in-office laser surgery on the vocal folds, the pool of people who can receive this treatment becomes larger and more people are able to save time and money. So thank you for listening. Next, I'm going to pass it off to Professor Petke.
Hi. Rebecca, I think you need to allow me to turn my video on because I forgot to turn it off in the beginning.
Sorry about that, Doug. Hold on one second.
No bad, my mistake in the beginning. Perfect. I forgot to turn it off in the beginning, so that was the glitch. So those were absolutely phenomenal, right? there's not too much I can say at this point that adds to this. And I think in the beginning I told you this was going to be the best event of Arts and Sciences Week, and I clearly stand by that. The talks were phenomenal, and I want to thank them for all of their hard work and the support networks that allowed them to do this work. The faculty labs they're in, the other students, the funding agencies, and the generous donors for a lot of the programs that help that. At this time of year, as we race towards project presentation day, graduation, planning for next year, our stress levels start to go up. And I just get so inspired by listening to these talks that it clearly makes it all worth it. And the only thing I have to say is I'm going to request Zoom to create a standing ovation emoji, because those talks were outstanding. All these projects clearly show the impact the students and WPI is making in the world to make our lives better, through research, towards making us healthier rather than surgery or invasive procedures, through changing habits of diet, practicing mindfulness, or developing new technologies that are less invasive, as well as creating a sustainable environment and leveraging the creativeness of all peoples in the world. And so I want to thank you for listening to these great talks. Enjoy the upcoming activities. There are faculty lightning talks on Friday, and there's a video that you can watch in a discussion this afternoon on a virtual screening of coded bias, along with a discussion session. I believe it's at 3 o'clock this afternoon. And so with that, I would like to thank everyone and close out this session.